Hey all, welcome to my channel. Do like, share, and subscribe.
So here the next topic is digestion. Digestion is nothing but conversion of complex molecules into the simpler ones to make it easy for the absorption assimilation of nutrients from the diet we eat for the human body. To be very specific, we are going to talk about protein digestion. Proteins are nothing but a long chain of amino acids which are linked by peptide linkages. So, these proteins, whenever we are talking about the digestion, that means we are only talking about how these long chain of amino acids will be broken down to a simpler form of the amino acids to make it absorbable to our human body. So there are a lots of enzymes which are required for protein digestion. So the enzymes required for protein digestions are of two types: proteases and peptidases. So these proteases and peptidases are two types of enzymes required for protein digestion, and they in an inactive form zymogens are usually the enzymes which are in an inactive form, and they will be activated whenever needed and at an optimal pH. Now, talking about the flow of protein digestion, proteins will be first converted to proteoses, then peptones, then polypeptides, then dipeptides, finally converted into a simpler form of an amino acids, which is easily absorbable in the human body. Talking about the location where, like the parts where the protein digestion actually takes place, whereas in buccal cavity like in mouth there will be no protein digestion as there are no proteolytic enzymes present in the mouth. They are present in the stomach because of the high acidic content. So coming to the stomach, you can see here a specific enzyme which is helping out for protein digestion is pepsinogen, which is in an inactive form, in a zymogenic form. This pepsinogen will be converted to active pepsin in the presence of strong acidic pH, that is about 1 or 2 pH. This pepsinogen will be converted to pepsin. Now, this active form of pepsin enzyme will work onto the complex form of proteins that enters into the stomach, and then they hydrolyze to proteoses or peptones. So that means these proteoses and peptones are formed in the stomach itself by an enzyme pepsin. This pepsin in the stomach, they also hydrolyzes milk protein, that is casein, to paracasein, and this paracasein will be precipitated as calcium paracaseinate, and this is which what forms clotting or curdling of milk in infants. And there is another enzyme, that is prorenin. So this is an inactive form of enzyme which activate renin, and this renin in turn converts the casein into paracasein. So this prorenin and renin usually present in infants and in calves as they intake a high amount of milk, and they are absent in adults. This pepsin, as I told you, it is highly active at pH, acidic pH, that is 1 and 2. They cannot withstand the alkalinity. So this this pepsin will be only present in the stomach, and they cannot enter into the intestine because of the alteration in the pH. Moving forward to the next part of the protein digestion after stomach is intestine. So in intestine, there are so many like different parts which involve in protein digestion in the intestine, and there are most part of the protein will be usually or principally digested in the intestine itself. So there are juices from the pancreas, from the intestine itself, and from the bile, the like the vicinity or the visceral organs, which releases all the enzymes in the intestine, so proteins can easily get digested. So these enzymes can be of two types: pancreatic proteoses and intestinal proteoses. Coming to the pancreatic proteoses, you have three different types of enzymes, that is trypsinogen, chymotrypsinogen, and procarboxypeptidases. So this trypsinogen, all these enzymes are usually called as enterokinases. Enterokinases are nothing but they are present in the intestine, in the gut. So these enzymes are actually in an zymogenic form, and they will be usually converted to trypsin and chymotrypsin and carboxypeptidases, and they act upon accordingly, and usually they work on the peptones and the proteoses coming from the stomach, and these proteoses and the peptones by the action of chymotrypsin, trypsin and the carboxypeptidases will be then converted to polypeptides and dipeptides. So these polypeptides and dipeptides will be completely getting digested in a second part of the intestine, that is the intestinal proteosis. Part where the complete protein digestion takes place is the intestinal proteases, where finally the complete protein digestion takes place, and the enzymes involved in this here is enteropeptidases, aminopeptidases and dipeptidases. So the complete protein hydrolysis starts by so many other proteolytic enzymes apart with these three, and they are active at a pH of 7 to 8. The main action of enteropeptidase is to activate the inactive trypsinogen into active form, and aminopeptidases, as the name itself says, they hydrolyzes polypeptides by removing amino acids which contains free amino group, and then the dipeptides are finally hydrolyzed into the amino acids.
This amino acid formation is usually the simpler form which is easily absorbed and assimilated by human body, and this is the end of protein digestion in the intestine. So that means protein digestion starts from the stomach and ends up in the intestine, and then from there the simpler form of amino acids will be absorbed and assimilated in the blood and the necessary parts. Thank you. If you have any doubts, you can comment down, like, subscribe and share.